Eric.
Andrew. Hey, Andrew, during the talk, can you just monitor the chat in case anything comes up?
Sure.
Any questions? Just so I don't miss somebody.
And you want the questions during the talk? Like
I'm happy to take them whenever. So
okay,
yeah.
Perfect. Just give it another minute, Eric, and then I'll introduce you. Okay, so we're a minute after, might as well get started. Thank you everyone for attending. Just some quick housekeeping. There will be also a seminar next month. Andrew Nikolowski in Biostatistics will be presenting. Actually, maybe it won't be Andrew, but there will be another talk next month. So today, Eric Clambey, who's the director of the Flow Cytometry shared resource, is going to be speaking about the best practices in flow and mass cytometry-based research, and also touching on experimental design and analyses of these experiments. So Eric, we're excited to have you speak with our group again, and you go ahead and share your slides and get going.
Great. Andrew, can you hear me?
Yep, and the slides look good too.
Great, thank you. All right. Well, first I'd like to thank the BBSR for the opportunity to present on flow and mass cytometry-based research. As Andrew said, I'm Eric Clambey. I'm an assistant professor in anesthesiology, and I'm also the director of the Cancer Center Flow Cytometry Shared Resource. So today I'm hoping to convey to you how to get the most out of cytometry-based research in a rapidly changing field while avoiding mistakes. And I have no conflicts of interest to disclose. I will mention some products along the way, but that's not endorsement. Those are just my own personal opinions. Okay, so before I get into the details, I just wanted to point out that one really valuable resource on campus that you have access to is the Cancer Center Flow Cytometry Shared Resource. I'm the director of that. Christine Childs is the manager, and we have an amazing team with Lester, Dimitri, Christine, and Chris. We are highly experienced. We provide cutting-edge instruments for all the technologies that I'll discuss today. And we also provide extensive education and training opportunities. So please, if you hear things that you'd like to learn more about, I'm happy to take questions, but our team is amazing and will absolutely be able to help you. So today I'd like to start talking about fundamentals of cytometry-based research, then discuss common considerations that are applicable for both flow and mass cytometry. Then talk about the evolving landscape of cytometry-based research, including different flavors of flow cytometry as well as mass cytometry. And I would welcome questions in the chat or either during or after the talk. So first off, let's talk about the fundamentals of cytometry-based technologies. So, let's see here. Just trying to get rid of there. Okay. So, for those, just to make sure that everyone's on the same page here. So both flow cytometry and mass cytometry are two powerful single-cell technologies. Both of these technologies enable rapid quantitation of cells and the properties particularly focused on protein expression, where your samples are being collected at hundreds to thousands of cells per second. So the basic components or requirements for these assays are you have to have cells in a single cell suspension, so cells are dissociated. You then have detection reagents. In the context of flow cytometry, these are fluorescently labeled antibodies often. In the context of mass cytometry or CyTOF, these detection reagents are isotopically labeled reagents. And ultimately, what's being analyzed and read out in these different technologies, with flow cytometry, you're characterizing cell-associated fluorescence. So for instance, this fluorescent blue antibody is binding to this fluorescent blue protein, giving you, and so you're measuring the blue fluorescence as an indirect measure of that protein abundance on that cell. In contrast, mass cytometry or CyTOF quantifies cell-associated isotopic mass. So one reason why this technology is so incredibly powerful is that not only can this technology be applied to cells, but these formats have been extended to measure additional materials. For example, cytometry can also analyze exosomes, virus particles, bacteria. And while the detection reagents are frequently antibodies, there also are a number of additional probes that have been used. For instance, there are hybridization-based probes for RNA, as well as either fluorescent or isotopically labeled compounds that can bind to cells with interesting and important biological properties. So some example measurements of what these technologies can measure include cell death, cell division, changes in protein and RNA expression, reporter gene expression, as well as identifying cell types and cell identity. So there are hundreds of different ways in which you can use these technologies. A few common examples. In this top right panel, we're looking at a case in which cancer cells are treated with either a vehicle or a chemotherapy. And you can use these technologies to assess how individual cells are responding across that population. For instance, if there are cells which fail to respond, those in tan, versus those that respond to the chemotherapy. This can allow you to really understand if there might be resistant cell populations within the bulk population. You can also simultaneously measure multiple parameters. And this is really where the rubber hits the road with cytometry, because it allows you to define what processes are co-occurring within individual cells or are not co-occurring. So for instance, in this cartoon, the cell on the right-hand side is undergoing apoptosis and has a unique post-translational modification of a protein identified by this purple P. In contrast, this cell has induced a reporter gene as well as expression of a cell surface protein, in contrast to the cell on the left-hand side, which has a distinct protein phosphorylation event as well as distinct cell surface protein expression relative to these other two cell types. So imagine that you're applying this level of resolution to the above scenario, comparing response to a chemotherapy. These technologies are also frequently used to quantify cell type or cell identity, for example, in peripheral blood, but also in the tumor microenvironment, where you can assess the relative proportions of different leukocyte populations, whether neutrophils, monocytes, lymphocytes, epithelial cells, etc. One important limitation of these technologies is that the cells must be dissociated into a single cell suspension. So there's no spatial or tissue context. So two additional things I want to point out about this. For the vast majority of cytometry-based studies, antibodies are critical for rigor and reproducibility. If you have bad antibodies, you are at a significant disadvantage with these methods. But ultimately, these data provide single-cell quantitation of cell-associated labeled antibodies or probes, which either measure fluorescence or elemental isotopic mass. There are three broad technologies within the cytometry-based research field that I'll focus on today. The first is cell sorting, or fluorescence-activated cell sorting, or FACS. This technology purifies viable cells based on fluorescence. And so you have cells stained with different fluorescent molecules, and a cell sorter can actually purify populations based on their differential fluorescence. And these purified cells can then be used for downstream assays. Now, flow cytometry quantifies cell-associated fluorescence, and it's intimately related with FACS, okay? But a very important distinction here is that a flow cytometer, while it can collect, between 1 to 20 plus parameters per cell, collect thousands of events per second, ultimately, this is a terminal assay, and you do not recover cells using a conventional flow cytometer, in contrast to a cell sorter. Now, there are two types of flow cytometry, conventional and spectral flow cytometry that I'll discuss later on. And then the third technology is mass cytometry, or CyTOF, which again quantifies cell-associated isotopic signal. The real advantage of this, of CyTOF, is the ability to go extremely high dimensional. And these assays frequently are quantifying at least 35 parameters per cell. And while this instrument is slightly slower than a flow cytometer and a terminal assay, the wealth of data that you can get from CyTOF is frequently unparalleled. I also like to point out that sometimes people refer to flow cytometry as FACS, but please note that FACS is cell sorting and flow cytometry is the terminal assay, in which both measuring fluorescence. So today I'd like to talk about broadly basic steps in these studies and then provide additional detail along the way. So broadly, I've broken down cytometry-based research into 5 steps, from design setup to sample collection and sample processing, acquisition, and analysis. Now, critical factors for success include a robust experimental design, protocols, and reagents, identifying, controlling for, and mitigating variables. And I'll spend a lot of time on this because I think if you don't take that into consideration, your results can be significantly compromised. Obviously, you need high quality samples and standardized sample processing, as well as well-maintained instrumentation. I also would like to point out both the strengths of these technologies, but also to remind you about what these technologies are measuring and what they're not. So I already mentioned that they can't provide spatial context. I would also like to point out that in the process of obtaining a sample, for example, tumor tissue, ultimately, because cells need to be dissociated into a single cell suspension to be analyzed on a cytometer, there are a number of experimental and data processing steps along the way. These are inherent in the technology, but I'd like to point out that along the way, there are some points in which you can have either experimental or technical dropout of different cells and cell types. So for instance, if you have a tumor tissue, it has to undergo mechanical and enzymatic digestion, and you will get some dropout of cells and extracellular matrix with this process. You then typically filter this material, which will remove dead cells and cell clumps. The cells are stained with antibodies, and then put on the instrument. But along the way, you've already removed debris, cell clumps, dead cells. Once samples have gone through the instrument, you have data analysis. And there's a series of analytical steps for cleaning up your data. But suffice it to say that this involves focusing on cells based on their cell size and their scatter properties. But you also remove doublets, because since cytometry is a single cell focused technology, doublets have the possibility of confounding your results. You don't want to, for example, be very excited about an interesting cell phenotype, which is essentially just this orange cell and this green cell, which happen to be stuck together. You also remove dead cells in the analysis, and ultimately you obtain single cell data quantifying protein expression, which can then be aggregated into different cell types. But the point of this is that when you use these technologies, it is important to be aware of both the strengths and the limitations of them. I also would like to briefly mention possibilities of integrating or aligning cytometry imaging-based studies. So again, in this case of a lung tumor, if you if you seek to characterize that by cytometry, you will dissociate that tissue and then apply it onto the cytometer. In contrast to if you want to take, do immunohistochemistry. Now, there are some important points to consider if you are interested in trying to align these technologies. One is antibody performance. Many antibodies don't work across both platforms. And so it's important to realize or to be aware of your antibodies and their performance. You also need to be aware of the inherent differences in what you're analyzing. Often, cytometry is taking, for example, a biopsy where you dissociate that tissue and then run that through the instrument. And so there you're getting either that entire biopsy or you're getting an entire piece of tissue, which is then the entire tissue is being run through the instrument. In contrast, if you think about imaging-based studies, here you're using tissue slices and regions of interest. And as I already alluded to, you can have the impact of digestion for cytometry can result in differential cell representation. One case in which you might have a susceptible cell might not be as detected in cytometry as in imaging. It's also important to be aware of the assumptions and differences in the analysis. And one interesting strength of cytometry is because it is a single cell suspension, know that each event is an individual cell. In contrast with imaging technologies, you require additional things such as cell segmentation to identify individual cells, and given the close apposition of cells in situ, often you require additional algorithms to tease apart what proteins are expressed on what cells and neighboring cells. Okay. So let's talk about basic experimental design. All right, so the things that you need in your flow or mass cytometry experiments, obviously you need standardized sample collection and antibody staining protocols. You need your cells in suspension, your antibodies, your cytometer, and then you need controls. Okay, you need controls to define your background fluorescence or isotopic signal. You need controls to accurately assign your signal. Now, I can't emphasize enough, if you are doing flow cytometry, this requires controls. This requires samples stained with individual antibody fluorophores so that you can accurately interpret fluorescence and correct for artifacts. If you choose to do flow cytometry without controls, it's often impossible to correctly interpret your data. So I'll talk about this later, but if you are using a conventional flow cytometer, the primary use of those single stain controls is to apply compensation to your data to correct for artifacts in terms of signal bleed over between different detectors. For spectral flow cytometry, these controls are used for spectral unmixing. And of course, you might also need controls to define your antibody specificity. So on the next slide is an example of a typical flow cytometry experiment. So in this experiment, there are two groups, each with 5 samples per group. For example, a drug treated versus a not drug treated. And in this case, say you want to keep it pretty simple, you are just staying with 8 antibodies. And of these antibodies, 7 of those antibodies are robust. But there's one antibody against protein X that only gives a low or limited signal intensity. So what samples do you need? What controls do you need for this type of an experiment? Well, your core set of controls, which is essential, include an unstained control to define your background signal, your single antibody stain controls. So each color representing a different fluorescently labeled antibody. You can use beads or cells. And please note that depending upon your application, in some cases, beads might be preferable. In other cases, cells might be more preferable. Generally, you need to be aware of what signal, how brightly your antibody stain controls are, because you want to make sure that those single stain controls are at least as bright as your experimentally stained samples. Okay, then you'd have your experimentally stained samples. So here are your 10 samples stained with this cocktail of 8 different antibodies. So already here we have 9 controls. We then have 10 experimental samples. And then we have what I'll call experiment-specific controls. For instance, if you're concerned that protein X, if you want to have confidence that your in-house antibody against protein X is specific, you might stain with a knockout control, right? And so here you would stain with all 8 of the antibodies, but where the cells that are being stained have been genetically disrupted for protein X. Ideally, if your antibody is specific, you will have no antibody binding against protein X in the sample. It's also frequently important to measure the background fluorescence in a channel that measures, in this case, protein X. And so a common practice is to use what's called a full -1 or an FMO control, in which you have an experimental sample with 7 antibodies, but you've removed specifically an antibody against protein X. Okay, so in total, in this example, where there are 10 experimental samples using 8 antibodies, you would have 11 controls and 10 experimental samples. Okay. So in terms of considerations for design and setup, the first thing I'd like to point out is things which are inherent across technologies, but I still think are worth emphasizing because it's easy. I've run into many people who have overlooked them. So first, the scale of your experiment really profoundly influences the experimental design. So what do I mean by that? Well, if you're looking at a single time point, in a cell culture, or multiple time points, if you're doing a time course in cells or in patients, or if you're doing a large cohort study, with each of these different levels of sophistication, it requires additional levels of controls to make sure that you are obtaining consistent signals, you're measuring consistent signals across time points, and that your analysis is not subject to batch variation. For example, that experiment was done on Monday, that experiment's done on Thursday. Oh, and actually the major variation between them is simply because there are some experimental variable that differ between Monday and Thursday. This sounds like a no-brainer, but be sure to sufficiently power your study. Be aware of the dynamic range of the process you're studying, and make sure that you have powered the study sufficiently to find the differences that you anticipate might be present in your experiment. It's also critical to define and mitigate variables that can impact your study. That includes having standardized sample processing, controlling for antibody variation. For example, you don't want it to be changing antibody lots between repeat experiments or across a time course. Two methods that can facilitate or can address some of these experimental variables include the use of internal standards, as well as the possible use of barcoding, a method that is especially prevalent in mass cytometry or CyTOF, but is also possible in flow cytometry. As I mentioned, you need to optimize sample collection to ensure high quality, consistent samples. And there's a significant amount of time involved in optimizing, refining your antibodies and your staining protocols. That includes, which antibody clone you're using, antibody titration, and panel design, which I'll talk about a little bit later. So antibodies are a foundation of cytometry-based research and are critical for rigor and reproducibility. It's possible to use both monoclonal and polyclonal antibodies, although monoclonal antibodies are by far the more common. Questions that you should ask when you're considering using antibodies for flow cytometry. First, is the antibody you're using, is it intended or validated for cytometry-based research? There are plenty of antibodies which, they might work on a Western blot, but whether they will have any performance in the context of cell staining and cells in suspension that needs to be verified. Of course, you need to have evidence for antibody specificity and validation, whether commercial through the vendor or in your own lab. You also need to be cognizant of how the antibody is detected. Is it directly labeled, or are you using a secondary detection reagent? So, for example, an antibody that's then detected by a secondary antibody. You should also pay attention to the recommended antibody staining protocol and what buffers they recommend. This is particularly relevant in the context of intracellular stains, but there are many antibodies which are used for intracellular staining, and you need to pay close attention to what buffers the companies are recommending to use. For example, some companies will often use a very harsh fixation such as methanol, others use a milder fixation. But the reason that matters is that those staining conditions can profoundly influence your other antibody performance. And of course, you need to define your optimal titration. So in terms of antibody use, antibodies need to be titrated to achieve an optimal signal-to-noise ratio. This is true across disciplines in biology, but here's just simply an example of a dilution series of an antibody on cells, where the positive population is at the top and the negative is at the bottom. And you want to find that sweet spot where if you use too much antibody, for example, over here, you have a significant increase in the background signal with potential spillover into neighboring detectors, for example, on conventional flow cytometry. If you use too little antibody, you don't have optimal separation. And so there are various approaches toward defining the optimal antibody titration, generally focusing on a separation concentration, which allows robust distinction between the positives and negatives. But you can also use numeric values such as a stain index, which are present in many flow cytometry software packages. It's also worth noting that for flow cytometry, the fluorophore that you use can profoundly influence the result. Fluorophores differ in terms of their brightness, stability, what laser they're excited by or are not excited by, and the resulting light released from that fluorophore. I think one critical concept here is that the exact same specificity of an antibody, if it's conjugated to two different fluorescent molecules, can give you very different measures. So if you compare the top versus this top blue arrow versus this lower blue arrow, here, this is an extremely bright fluorophore, and all these different analyses here are with the same antibody, but you can see a very large distinction between your positive and negatives. In contrast, the same antibody specificity simply conjugated to a different fluorescent molecule down here, while you still have a positive and negative, the difference between those is greatly reduced. Okay. So why does this matter? Well, it matters because you need to try to pair the antibody and the fluorophore with the known characteristics of your protein target. If you have a protein that's expressed at a low level, these are targets in which you want to use an antibody conjugated to a bright fluorophore that will really provide optimal detection. If you are trying to stain for a protein expressed at a high level, these can be detected using weaker fluorophores. Okay, so a highly expressed protein, for example, you could get by with this lower signal-to-noise ratio fluorophore, but you would not want to use this dim fluorophore to detect a low abundance protein. Now, where this really becomes an issue is when you're measuring more than one parameter, and that's what the vast majority of us want to do. And so here, panel design is key. And what panel design refers to is designing the optimal pairing of antibodies and fluorophores to balance fluorescent signals and to minimize technical artifacts. Okay. So in terms of antibody fluorophore or isotope conjugates, which antibody and fluorophore you're using, for example, is influenced by the machine you're using, what lasers and what detectors does the instrument have. If your instrument only has 3 lasers, some fluorophores are not going to be excited on that instrument and would be of no value. You need to think about how many antibodies you're simultaneously measuring. You need to optimize the combination of fluorophores to get this best signal-to-noise ratio and to minimize artifacts. For CyTOF, there is some variation in signal intensity across isotopes. It's not as dramatic as the dynamic range that I showed for the fluorophores, but that can also inform what isotopes you would use for different targets. And as I mentioned, antibodies need to be titered. So in terms of collection, please be aware of how your sample collection affects downstream results. Okay, this includes, whether you're running your samples fresh. Sometimes that's possible. More often in the context of animal models, or whether you're using frozen material. How you're freezing those samples is critical, and the consequences of enzymatic or mechanical processing. Also, one important context in which you can get different results is if you're using PBMCs versus whole blood. So for instance, PBMCs, that process removes neutrophils, whereas in whole blood analysis, you can detect neutrophils. And obviously, garbage in, garbage out. If you have poor sample quality, that is often the death of your experiment. In terms of sample processing, please be aware that not all cells tolerate dissociation equally well. So if you are concerned that your cell might be fragile, certainly optimizing and assessing for optimal recovery of that cell from a tissue is important. It's also important to remember that the enzymes that we often use to digest tissue, they're cleaving proteins in extracellular matrix. And so you need to also quantify the impact of those enzymatic treatments on your protein expression. You also need to optimize your antibody staining and in particular, if you're using intracellular stains, make sure that the antibodies, the fluorophores, and the buffers are all compatible. For instance, if you stained with an antibody linked to PE, the fluorophore PE, on the cell surface, and then you come back and use a methanol for fixation, the methanol is going to completely destroy the structure of PE, and you will obliterate your PE-associated fluorescence. And so there's a lot of literature in terms of knowing fluorophores that are compatible or incompatible, but please be aware of that. In terms of acquisition, obviously your instrument should be routinely calibrated. Please also note that the same instrument with the same settings will not be identical from day to day. Now, there are things that you can do to better control for that, but inherently these are instruments with variation. And then I can't emphasize enough, controls during acquisition are critical. You need those single stain controls to accurately interpret your fluorescence. and whether compensation or unmixing. And I see that there's a couple questions there. Okay, all right, great. All right. So regarding analysis, and I'll go into more depth on this, but analysis requires signal correction. I can't emphasize this enough. Now, there are different types of signal correction depending on the technology. Compensation is commonly applied for conventional flow cytometry. Unmixing is commonly used for spectral flow cytometry, and normalization is commonly used for CyTOF. Your data can be analyzed using multiple software algorithms. Some common commercial opportunities include FlowJo, FCS Express, Kaluza, Cell Engine. This is not exhaustive. These are just some common ones. And please note there are both manual and automated opportunities for analysis. Now, I think one of the really exciting but also challenging things is that cytometry-based research continues to evolve. So over the past decade, there's been a major push towards higher dimension analysis. And this has really been powered by two things. One, increased instrumentation, advances in instrumentation, and two, expanded reagents. So there are now, there continue to be more and more fluorophores with unique fluorescent properties identified, which really allow you to expand this. So for instance, last year at the annual Cyto conference, there was someone who gave a talk, and they were measuring, I think that they actually had managed to measure 70 parameters by flow cytometry. I mean, it was a Herculean task. And certainly that's well beyond, the vast majority of labs. But suffice to say, like the field continues to push towards higher and higher dimensions. So why does this matter? Well, let's talk about conventional versus spectral flow cytometry and mass cytometry. So as I mentioned before, flow cytometry relies on fluorescent detection. Fluorescent molecules emit light across a range of wavelengths. So for instance, if you focus on PE-Cy7 here, what you're looking at is the emission of this fluorophore, PE-Cy7, with wavelengths on the x-axis. Here, they're looking at fluorophores that are excited by a 488 nanometer laser. But what you can see here is that there's a breadth of fluorescence of light emitted from a PE-Cy7 molecule. If you look at along the spectrum, you can also see that fluorophores, many of these fluorophores have overlapping emission. Okay, and so for instance, if you want to have confidence that an antibody detecting label with PE-Cy7 is that you can differentiate that from an antibody labeled with Per-CP-Cy5.5 or Per-CP, you need those controls to deconvolute or to compensate to accurately assign your fluorescence to the individual fluorophore. And the fluorophore, remember, maps to that antibody and that protein that you're detecting, and the protein that you're detecting. Also, please note that different fluorophores are excited by different lasers. So for instance, APC Alexa Fluor 647 are excited by a 640 nanometer laser and poorly excited by other lasers. Okay, so let's talk about conventional versus spectral flow cytometry. So what's important to note is both of these technologies measure cell-associated fluorescence. They use multiple lasers to excite the fluorophores, and they both require controls. But there's an important difference here in terms of how they measure and how they assign that fluorescent signal to your antibody and your protein target. Okay. So conventional flow cytometry, as the name implies, has been around much longer than spectral flow cytometry. And the basis of this technology is that the instruments have multiple detectors that detect a range that detect light emission from fluorophores. Here, for example, this gray box is a detector that detects light emitted between approximately 420 to 500 nanometer. Okay. Now, the way that a conventional flow cytometer assigns fluorescence to an individual antibody is it's just focused on the light emitted in this narrow band of wavelengths. So here, one detector equals one color or fluorophore, and it's this narrow window emission identified with the gray box. In contrast, the more recently developed technology, spectral flow cytometry, again, it still measures fluorescence, but here rather than focus having a detector with where you assign that one detector to the one fluorophore, here each fluorophore is characterized by its full emission, that is its spectral fingerprint. So on the top panel, this shows the fluorescence of two antibodies conjugated to two different fluorophores that are closely related, and here they're virtually indistinguishable. But if you look on the bottom, you can actually see if basically on the x-axis are different wavelengths of light emission and different lasers. And the bottom line is that with spectral flow cytometry, you can often find differences between fluorophores by not only looking at a narrow length of emission, but across the entire wavelength spectrum. So there are still many conventional flow cytometers around, as well as spectral flow cytometers. I would say that in general, there's been a real enthusiasm and burst for the spectral flow cytometry, given some of its unique strengths over conventional cytometry. But yes, so for conventional flow cytometry, it was the original technology. It's limited by the breadth of fluorescent emissions. It cannot readily discriminate between closely related fluorophores. In contrast, spectral flow cytometry differentiates based across the spectral fingerprint across the entire emission spectrum. And it allows expanded detection, including fluorophores, which might be difficult to differentiate using conventional. So why does this matter? Well, the way that you handle data processing varies between these platforms. Okay? And so you should be aware of the strengths and limitations of these two. So with conventional flow cytometry, the challenge is that fluorophores have broad emission. Okay, and one fluorophore can be detected in a neighboring channel. And so for this, you use compensation. Now, compensation relies on you collecting those single stain samples, sample stain with individual fluorophores, as I mentioned before. Now, with spectral flow cytometry, the challenge is that each fluorophore has a distinct fingerprint, and you have to have all those distinct fingerprints because the algorithm that's used is called spectral unmixing, which deconvolutes the individual emission profiles. But obviously, if you don't have each spectral fingerprint for each fluorophore, you are not going to be able the machine isn't then, and the software is not going to be able to unmix and specifically assign the fluorophore of interest to your accurately. Okay. So the take home here is both these technologies measure cell associated fluorescence. They both require those single antibody stain samples, while conventional flow cytometry uses compensation to do signal correction. Spectral primarily uses spectral unmixing, but both of them, you have to have those controls. Now, what about mass cytometry or CyTOF? So again, this technology, the cells are in suspension, but the difference here is that rather than measuring fluorescence, you are measuring antibodies conjugated to isotopically purified rare earth lanthanides. Okay, but you can readily measure more than 35 to 40 parameters per cell. And the way that this works is that your antibodies are bound with isotopically purified elements. And so, for example, you can actually differentiate between europium-151 and europium-153, and those can be bound to different antibodies to allow you to differentiate between protein abundance with those two, measured by quantifying isotopic mass. Now, one really important difference between mass cytometry and flow cytometry is the breadth of the signal. Remember, with flow cytometry, I pointed out kind of the breadth of those emission wavelengths. In contrast, with mass cytometry or CyTOF, you're simply measuring mass. There is no breadth of mass, right? It's an absolute value. And so the cross-channel artifacts in mass cytometry are much less common. That's not to say that there are none. But they're relatively minor compared to flow cytometry. And those artifacts come from isotopic impurities and from oxidation. Okay. Practically, given the number of parameters often measured in a CyTOF experiment, these experiments often do not include single stain controls. And so that's one one major difference between doing a cytoph experiment and a flow cytometry experiment. That's not to say that there would be no value in including single stain controls, but the requirement for compensation is greatly reduced in cytoph experiments. I'd just like to point out that with cytoph data, one of the important differences with this technology is that you actually normalize the signal to internal equilibration beads as an important part of the analysis pipeline. Okay, so let's talk about data analysis, compensation, and visualization. So just a reminder, what primary cytometry data look like is often something like this, where an individual dot is data from an individual cell. And in this map, there's a heat map that indicates, when there are multiple cells at the same physical location on the map, it's a greater density. So here, red. Okay. So one of the things you'll often hear about in flow cytometry is gating. And so what's gating? Well, it's simply a method that allows you to focus your analysis on the cells that you care about with the characteristics that you want to look at, and it allows you to ignore other cells. And gating is typically done, there's a lot of post-acquisition analysis. What this data analysis will look like if you're using gating. In this top left panel, this is showing all the cells from a peripheral blood sample, for example. And if you want to query the expression of different proteins on lymphocytes, which have smaller cell size or forward scatter, and smaller scatter or granularity properties or side scatter. In flow cytometry software, you can draw a polygon around this and tell the software, I only want to look at those cells. And when you do that, this panel is allowing you to only focus your attention on the green cells here, and then querying expression for CD3 on cell surface protein on the x-axis versus CD19. And again, you can iteratively do this where you're saying, now I want to only look at cells that express CD3, but do not express CD19, and I want to assess their protein expression for other parameters. And so this gating allows you to sequentially focus your attention on specific subsets of cells. Now, one question is, how do you know where to draw a gate? Okay, this is pretty straightforward. Okay, clearly a negative versus a positive population. But what about in the case of something like this, right, where there's kind of a smear? There's no clear distinction between your positives and negatives. So here, this is really where you need your controls. Okay, whether you have a sample that's a knockout for your protein target, or whether you have a full -1 control where you can basically drop out this antibody and then you can define the background fluorescence. Those kinds of controls will allow you to assign the specific fluorescence to that antibody and that protein. And without those, some of these can be much more difficult or much more judgment calls. Now, one primer that I've found to be very helpful over the years is a review from the Herzenbergs. Although it's a bit dated now, I think some of the fundamental principles that they detail in this article is really helpful. So let me just talk through a little bit of why you need compensation and how that can influence your results. And so here, I've already alluded to the fact that when you have neighboring fluorophores, basically you can get the emission to overlap. And so in this sample, on this top right plot here, the sample was only stained with an antibody in the green channel on the x-axis. And yet, if you look, these cells appear to be double positive, but they're not double positive because experimentally, the only thing that they were stained with was one antibody. And so compensation allows you to mathematically account for this detection in the neighboring detector, and then allows you to bring these two samples so that there is no longer spurious or artifactual signal in the neighboring channel. But please note, even correctly compensated or unmixed data, you can get signal spread. So here's the negative population. If you look at the positive population, it's not a tight circle. Instead, there is some spread, and signal spread is one challenge when designing a panel with multiple targets. I also would like to point out that how you visualize your data can profoundly influence your interpretation. So this is a log 10 plot, but you can see many of these events are buried on the axis. And if you use instead something like a bi-exponential plot, which allows you to deal with both kind of logarithmic values as well as kind of compressed values around 0, that can also allow you to better visualize your data. Okay, so let's talk a little bit more about high dimensional analysis. What do you need to consider? Well, one, you need to think about panel design. This requires really optimizing your antibodies, your fluorophores with your protein targets, right? Are you making sure that you're using dim fluorophores with highly expressed proteins and vice versa? And this can be a daunting task, the panel design. And what I'm talking about here is, I mean, panel design is relevant for any number of parameters, but especially when you're getting high dimensional 15, 20, 25 parameters, that's really where it can become very challenging. So if you are going to want to make up your own panel, I would encourage you to talk to, core staff, whether it's ours or other flow cytometry cores on campus. I also would encourage you, companies are aware of the difficulties of panel design, and every company that has gotten into high dimensional space is offering something where they have technical specialists who can assist you in panel design. I can't emphasize enough. I would definitely encourage you to reach out to people and not try to do this by yourself. I would also ask if you can consider making use of a pre-designed panel. So for instance, there are published, well-established panels called OMIPs, or optimized multicolor immunofluorescent panels, against a number of things. And there also are now an increasing number of commercially available high dimension kits. I also think these are a great opportunity to really get into high dimensional analysis. SciTech and Standard Biosciences both have these high dimension kits. And I think they're only going to continue to come online. And I would strongly encourage you to consider those, because these are really optimized to work out all the kinks and to save you a lot of time if they can meet your experimental needs. I would also emphasize you need to control for variables. You need to make sure that you have consistent antibody performance. And I would strongly encourage you to include reference samples and possibly sample barcoding to address variation between samples and between runs. Now, the analysis is often more challenging with high dimensional data. And where to start there? Well, the good news is that there are now a number of algorithms that have been integrated with commercially available cytometry software, such as FlowJo has now a whole series of plugins. I would also point out that there are fee-for-service analysis options available. Astrolabe Diagnostics was founded as a CyTOF analysis company, and they have incredible skills and ability to analyze complex data sets rapidly. The other thing to think about for high dimensional analysis is, when I gave examples of gating in terms of like a Boolean analysis of, X versus Y and A versus B, if you think about 20 or 30 parameters, this can become extremely arduous. And so there's been a move towards data analysis for high dimensional analysis to really integrate two different aspects of two different approaches. One is dimensionality reduction, and the other is clustering. And these are frequently integrated together to allow robust visualization. And there are a variety of algorithms that are out there. What's important to note is that some of these algorithms are really focused on single cell resolution, others at cluster level. Some provide the ability to look at individual samples, others provide an aggregated analysis. One thing I would really recommend is if your data are visualized at the cluster level, please take the time to query the data at the single cell level to make sure that you really understand how homogeneous or heterogeneous you are. I would also say there is not a single magic algorithm. And for instance, this is already an out-of-date example, but there are numerous different algorithms identified with the gray boxes. And interestingly, the data shown here is the exact same data set, okay? And so this simply exemplifies that different algorithms are visualizing the data differently. And you need to be conscious of what the different visualizations depict. And if you are interested in some examples of this, I would strongly encourage you to look at this paper that Abby Kimball and my group previously published, which walks you through some of some of the data visualization and considerations for these algorithms. Okay, so these algorithms are really influenced by a number of parameters, and these include those on the left-hand side here. But please be aware of what's of this if you are getting into high-dimensional algorithms. What's your input population? Are you putting in all cells or all viable cells? Are you putting in a pre-selected population? Because depending on what you're putting in here, you can get very different results. The results are also influenced by the number of cells in your input. But if you're clustering, are you clustering on all parameters or only on some? You need to be aware of the algorithm settings, and also just be aware of stability and reproducibility. Whenever we use these algorithms, we keep records of the input population, the parameters used for clustering, and the algorithm settings. Okay, so ultimately, the goal here is for you to have confidence in your cytometry studies. So obviously you need to use well-calibrated instruments. You need to optimize your antibodies and have robust samples. I can't emphasize enough, you have to have controls, okay? Those single stent controls to allow for signal correction for flow cytometry are essential. And then there are a variety of negative controls you can include, such as knockout cells, a full -1 control or FMO. If you are going to use isotype controls, please be aware that isotype controls have been selected to give minimal binding signals. And so there are some extra considerations you should be aware of if you really are relying on isotype controls. Kind of in terms of big pictures, the one thing I want to emphasize is that robust flow and mass cytometry really begins with strong experimental design. You end up spending so much time and money on these types of studies that I can't urge you enough to really think critically in terms of your experimental design, what you can do to control for variables, how you can design robust experiments that are going to give you reliable results. This includes investing in optimized sample processing and staining, really nailing your antibody panels, including all your controls to obtain meaningful data. I would also emphasize, when you are sharing your data, be transparent in terms of how you've analyzed your data, what cell populations you present, when you publish them, and just be aware that your data analysis and visualization can heavily influence your interpretation. And so, whenever possible, I try to use complementary approaches or complementary algorithms to make sure that it's not simply that one magic way that I visualize the data in which I can see a population. So I'd like to thank you for your attention. I'm happy to take questions now or later. And certainly we as the Kent Center Shared Resource are happy to answer questions further as well. Thank you.
Thank you, Eric. That was great. Are there any questions from the group? Maybe we'll get some in a second. Eric, what's the best way to contact your core?
Oh, sure. Yeah. Good point. I didn't include that on it. I'm sorry. It's CC Flow Cyto. I'll put it in the chat. Okay. Sorry. Yeah.
And I had a question about how the acquisition portion goes. Do you train people to run the machines themselves, or are all the machines run by your group, or is there kind of a mixture?
Yeah. So we have many people who we train to run the instruments, and they run them themselves. That's something we prioritize. We want to make sure that everyone has the ability to run these instruments, especially the cell analyzers. For cell sorters, we have one cell sorter that can be run by a trained individual. For the CyTOF, thus far, that piece of equipment is requires a lot of care. And so thus far it's been staff operated, but we are looking into options. There's a next generation instrument. And with that instrument, it's been designed to be much more investigator friendly. Yeah.
Okay, so yeah, the contact email is in chat now.
Thank you, Christine.
Does anyone else have any questions?
And really, we would, we love talking about cytometry. We would love, I love talking about it. Our team loves talking about it. And please, if there's something that wasn't clear, or you just have a question that I didn't even touch on, there's a lot in this field. And I think we would love to support you and make your experiments really sing. So.
Okay, well, thank you again, Eric. That was a great talk as always. And we will have another seminar series next month.
Thanks, everybody. Really appreciate it.